Have you ever looked at a massive forest and wondered how many plant species are actually in there, and how many each of them? And yes, you could try counting them one by one, but unless you want to have a happy life, you're probably gonna lose your mind before finishing the first acre. And that's why scientists developed a method to simplify the process known as the Quadrat Method. To show you how it works, I'm gonna start by measuring the playground where I'll do the experiment at. But I can just guess the size of this place by looking at it, can I? That's why I'm gonna use this map I got from Subnautica wiki and importing it to a technical tool called ImageJ. And the first thing to be done is that I need to set a scale from this tip in the bulb zone all the way down to the ground reef. And how exactly do I know how far is it from top to bottom? Well, I measured it directly, and it's 3,728 meters. So, I'm gonna put it in km, therefore 3.73. Okay, the measurement can finally begin, and I'm gonna use the freehand mode here to trace the irregular border of the map like this. Well, here it is, 0.3 square kilometer. Or for you who don't know, it's roughly the size of New York Central Park. Anyway, across that entire area, I found at least 19 sites that meet my requirements to do the experiment at. But I only needed 3, and choosing the correct ones is crucial here, because a single biased location can ruin the entire downstream results. So, to keep it scientifically valid, I need a sophisticated tool used widely by scientists as a standard in avoiding bias. This one. Well, submit then, site 10, 7, and 5. So without further ado, let's get out of this place and head directly to the first location, site 7. But before I start, I need to build something what ecologists call the plots. What is it, you may ask? Think of it as eating a slice of cake. From that one slice, you can imagine what the remaining of the cake tastes like without munching the diabetes out of it. And that's basically what plots are. And the first step of making them is to build a 4x4 meter rectangle, and in this case I'll use the power lines. Now the rule is, if adding a second plot next to it with the exact same size increases the species count more than 10%, it means the biome is still hiding some exciting things, hence a third plot becomes necessary. And the pattern goes on forever until the increase becomes insignificant. So enough chit chat, I'm gonna start building it. Oh my god, it took me hours to build. I didn't know the power line system would be so complex. And yeah, by the way, those are platforms that I'm using there as a reference since they're more or less 8 meters wide, which will be perfect for my 4 meter-ish plots. Okay, now let's do the cataloging step. In the first plot here, my eyes were caught immediately by none other than renowned acid mushroom, followed by 2 vine needles, 1 wreathing weed, and 2 twig-looking corals, which I'm just gonna call them the twig. Moving on to the second plot, we've got some familiar faces here from the previous plot, minus the nettle. But interestingly, they are replaced by 2 new exciting newcomers, the blue palm and the coral shell plates. Anyway, to the third plot, I am directly greeted by 2 distinct species of seaweeds here. I call the first one green round bush, and the second one green diamond bush, adding new collections to my data. And of course, these 3. Finally, the last plot reveals 6 distinct species, but unfortunately, since there's no new ones, the count fails to reach the 10% threshold. Hence the plots stop here, ending what we call the species area curve. Anyway, so what are these numbers for? And what do they mean? To answer that question, I've gotta go back to my thinking chair first.
Welcome aboard, Captain.
Oh, thanks. Okay, so, to make those previous data meaningful, we have to look up to 3 main parameters: density, frequency, and coverage. And by summing them all, we get something called the importance value index. The first one, density. This basically tells us how many individuals are there per square meter area, while frequency shows how often does each species appear across all the plots. Lastly, coverage is a little bit different because to get it, I must measure each of their surface area relative to the plot size, using ImageJ again. And to make a long story short, I insert those numbers into this formula to get this. And by the way, the numbers here are all their relative values since they're the one that can be used to compare to each others. Anyway, the results are in. It's very interesting to see how dynamic the result is. Where some species were found abundant in one side, they could be overtaken in the next one. Although, in the end, the acid mushroom is still a clear winner. And believe it or not, this game is actually full of scientific backups. As I was reading the mushroom's description, I found that this species actually has an extreme defense mechanism, releasing strong acid when scratched, and that's what may contribute to their high numbers. On the other hand, the least abundant species like this blue palm doesn't have any defense mechanism as we know it. Also, from the second list, the vine nettle, we know that they could be under constant predation by the grazers, which may explain their low abundance. Now moving on to the next biome, here light gets more and more scarce. You don't believe me? There. And it's literally in the middle of the day. Anyway, look to the front, what is it, Miss PDA?
Detecting volcanic activity and several unusual electromagnetic signatures in the region. Exercise caution when diving.
Precisely. So, although light becomes more and more valuable here, this biome is fortunate enough to be blessed by hydrothermal vents. They spit out thermal energy and mineral-rich chemicals. This is what fertilized the biome actually. And despite having size double than that of the spawn-safe shallows, I only found 17 proper sites here, mostly for the uneven terrain. And yet, it didn't stop me from running the method all over again from obtaining site 14, 13, and 4. Also, on the contrary of needing 4 plots in the safe shallows, I only needed 2 for this biome. The reason? I already found all the 3 species in the first spot. And these are their analysis results in average from 3 sites. Okay, now it is time to dig out what secrets lie hidden within those local residents. First, I already mentioned that underwater volcanoes fertilize this biome, right? Yet at the same time, they increase the environmental temperature. So, the first thing you need to have to adapt is heat tolerance. Moreover, from the bulb bush data here, we can see that one of the adaptations for this place is to have deep root system, anchoring the bodies to the terrain they're at. But here where it gets fascinating because it turns out there are delicious meal to herbivores in the biome. Even so, those deep roots alone could grow back into a new complete organism. And not only that, because when we hit a grown bulb bush, it will drop fragments instead of seeds, unlike other plants. And yet again, plant those fragments in the growing bed, you'll get full individuals. So what's going on here? From what we know so far, it seems that this plant possesses what here on Earth we know as totipotency, an ability to grow organs, tissues, and even cells to a whole new complete bodies. Cool, right? Furthermore, this biome also accurately depicts another kind of adaptation for deep seas, where the other 2 successful species are colored reddish. But why? Apparently, oceans filter out most of the red light, as it is the first to disappear in deep seas. There, having red-colored body makes you practically invisible to predators, as they see you of having gray or even black body. Okay, so far we have distinguished 2 types of biodiversity that rely on sunlight and deep sea vents. And in a biome, there has to be at least one group classified as autotroph, organisms that can make their own food, the chef of the seas. And the raw ingredients are sunlight for photoautotrophs and simple chemicals for chemoautotrophs. And now, I have arrived at the last biome to complete the whole picture, the sparse reef. Here everything is...
Diversity in this region is unusually low. Cause unknown.
Thank you, well, there it is answered just like that. And she was right, the diversity here is very low. Although she said she doesn't know the cause of it, I would bet on the light intensity and absence of active volcanoes. But yet again, I'm still thinking if there's an actual way to do the analysis. Considering the fact that it stands directly next to the ground reef that has dozens of fans, the current direction is heading towards it from the reef, luminance, elevations, etc. Well, I'll let you know if I figure it out in the next videos. From now, what I can say is the biodiversity in the sparse reef is sparse. Also, looking at the present flora, I found that this biome maintains the consistency of the adaptation for deep water, where pigmentations are still leaning towards the red color while some of them are possessing differentiated organs. For example, these photosensitive eyes. But the biggest mystery exists in this biome is actually the opposite of being unnoticed, these towering pillars. And I've been wondering what they are since the beginning of my gameplay. And actually I have a theory for it. First, I'm gonna do a quick disclaimer. Since I'm not a geologist, I'm not very familiar with geological activities. That being said, from my point of view, these spike-like structures actually resemble an exaggerated version of layered cementation of sediments. Here on Earth, we call them stromatolites. A structure formed by microbial activity such as cyanobacteria that hardens over time. And this planet takes it to another level. Look at those layers like pattern and not erosion-like, and see how they're actually cylindrical rather than striations along the cliff. Then notice how they're texturally different than the cliff's rock itself. Or maybe they're just another rocks. Well, here we are again watching one of the two things that give life to this planet. And on my journey of searching for ideas, I found that this planet has a great range of diversity that I hadn't noticed before. And that truly matters because in the end, diversity gives life resistance to disaster and to thrive. Finally, I hope you enjoyed the video and stay tuned for more biology behind Subnautica. Thank you for watching and bye!